This is Campbell's Biology, Chapter 17, Gene to Protein. So we're talking about how to convert DNA into protein and how genes get expressed, basically. That's what we're describing here. And it's two stages, transcription followed by translation. The translation portion is done by the ribosome. Here's a diagram, an image, a ribbon model of a ribosome. Small subunit on the bottom, large subunit on the top. So our DNA, we know, is segmented into various types or various parts. Some of that we call genes, and Archibald Gerald was the first one to identify or to suggest that one gene codes for one protein. This was in 1909. Pretty amazing. And then following that up, Beadle and Tatum worked with bread mold and X-rays, and they mutated the bread mold using the X-rays, and then hypothesized that there was a one-to-one relationship. One gene coded for one enzyme, and thus you could create a mutant that could block or not be able to complete an enzymatic pathway because it was defective in that one gene or one enzyme. They also worked on a specific pathway, the arginine pathway, and they used that arginine pathway to both identify or to consider the one gene, one protein, or one gene, one enzyme, and also the actual arginine pathway to confirm that. So here's their basic experiment. They worked with Neurospora, and they isolated mutants that required arginine to grow on in their growth medium. And these types fell into basically three types of mutants in the pathway. Because they believed that ornithine and citrulline were precursors to the production of arginine, which is an amino acid. This was their essential experiment. So the wild type strain required only minimal medium. So as you can see here, here's the wild type, this column, and whether you give it minimal medium or minimal medium with ornithine or citrulline or arginine, they all grow. You see that little orange stripe on top. Class 1 mutants, if you just give them minimal medium, they wouldn't grow. They would only grow if you gave them arginine or citrulline or ornithine. Class 2 mutants, class 3 mutants, same idea you can see. For class 3 mutants, you had to give them arginine. You couldn't give them citrulline or ornithine, they still wouldn't grow. So where is the mutation? So here's their basic results. There's 3 genes, one that codes for enzyme A, which is the enzyme that regulates the production of ornithine from this precursor. Gene B is from ornithine to citrulline, and gene C is citrulline to arginine. Now, class 1 mutants, they were mutated in gene A due to the X-rays, and as a result, you give them this precursor, they couldn't survive, they wouldn't grow. But if you give them ornithine, they would. Again, as you see here, these class 1 mutants, you give them just the control, they wouldn't grow, but you give them ornithine, and they do. Here you see you give them ornithine, and they grow. Class 2 mutants were mutant in gene B, and class gene C mutated, you had to give them arginine if they were going to grow. They couldn't use any of these other precursors in there. So this basically confirmed the arginine pathway and the one gene, one enzyme hypothesis that they had. And so the hypothesis is the function of a gene is to dictate the production of a specific enzyme, basically. So over time, we've made adjustments to this one gene, one enzyme hypothesis. It's not so simple because we do know that genes code for polypeptide chains as well as the intermediate RNA molecules, but there's also the potential for one gene to code for multiple polypeptide chains, and we'll show you how a little bit later. Now, DNA is converted into RNA. It's called transcription. And initially, we produce messenger RNA. And then translation takes that messenger RNA at the ribosomes and converts it into a polypeptide. In prokaryotes, transcription and translation occur together in the same location because they're not subdivided by membranes. They lack a nucleus, so mRNA is produced by transcription, and it's immediately translated into protein. And in fact, these can occur at the same time. While transcription is occurring, translation can occur on that RNA. In eukaryotes, on the other hand, RNA transcripts are produced in the nucleus, and then as they are being taken out of the nucleus, they must be modified. So in fact, when the messenger RNA is initially produced, we call it pre-mRNA, and it's not mature mRNA until it has a few modifications that we'll talk about here. This is the central dogma of biology, DNA to RNA to protein, which is what we're talking about. And we take the mRNA in the form of 3 nucleotides at a time, 3 ribonucleotides, that's ribosome, and that equals 1 codon. And 1 codon we know codes for 1 amino acid. So here we have DNA, gets transcribed into mRNA. Here's 1 codon, UGG. You look it up in the chart, and UGG codes for trip or tryptophan. So here's one amino acid in our long chain being coded for by these codons. Here's that chart that you look up. On the left, you have the first letter. On the top, you have the second letter. On the right, you have the third letter. So here we had UGG. If we look up UGG, we have U. U is the first letter, G is the second letter, and then G is the third letter. So here's UGG, and we have TRIP. You also notice a few of these code for stop. These code for release factors or stop factors that will end translation. And we also have a start codon, AUG, which codes for methionine, MET, methionine. Very important. All translation starts with AUG. So you have to read codons in the correct reading frame. You'll see later when we talk about mutations again that sometimes the reading frame can be shifted, and that causes serious problems. Every codon is changed. And we do know the genetic code is universal, meaning every single organism uses the genetic code, DNA and RNA, and as a result, we can take genes from one organism and transfer them to another. So here we have a fluorescing tobacco plant using a fluorescent gene, and here we have GFP, green fluorescent protein, put into I want to say that's a chinchilla, but it might be a mouse or a rat. Can't remember. But one species' DNA can get transferred to another and make transgenic organisms in lab experiments. RNA synthesis. So RNA polymerase is what takes DNA and makes it into RNA. And RNA polymerase is this giant enzyme, and it serves the purpose of all the other same or similar enzymes used in replication. That enzyme, they're all kind of part of this big complex that is RNA polymerase. So it pries the DNA strands apart, adds the RNA nucleotides, and then ends the process. And it's the same base pairing rules as DNA, except for U substitutes for T. We have uracil instead of thymine. The process goes by initiation, then elongation, and termination. And so here we see initiation, and here's the unit that we want to transcribe, this transcription unit or this gene. And upstream of the gene, we have a promoter region. And this is mainly eukaryotic transcription that we'll talk about here initially. This promoter region is where the RNA polymerase is going to bind, and RNA polymerase is going to unwind the strand and then add nucleotides one by one. And it does so, again, in the 5' to 3' direction. So it only does the leading strand. This mRNA is going to be single-stranded, and then it's going to fall off, and the original DNA is going to reanneal or reconnect back together. Okay, so here's initiation, promoter binding, elongation, adding the nucleotides or ribonucleotides, and then finally termination, the RNA transcript is released, and the polymerase falls off. Here you see a close-up picture. Direction of transcription is from left to right in the 5' to 3'. Again, we're adding nucleotides only to the 3' end there, and only on the leading strand, you can see. Now, I mentioned this word promoter. Promoters are found upstream or at the beginning of genes, and they're the region where the proteins will bind and initiate or start transcription. It's the signal to begin. About 25% to 1/3 of promoters also include a TATA box, which is the part at the beginning of the promoter where transcription factors begin to bind. Not all genes have TATA boxes, but some of them do, as I mentioned. And it's believed in part that they use T's and A's for the TATA box, and this is an evolutionary conserved sequence. They use T's and A's because they are the strength of the bond between T and A, only 2 hydrogen bonds, is weaker than G to C, so TA is easier to unwind than is the other types of sequences, G's and C's and a mixture. So here we have TATA box with transcription factors bound. Then that signals where RNA polymerase should bind. Here we have RNA polymerase 2. And it binds at those transcription factors over the promoter. And then we have initiation and elongation, as you know. And the RNA polymerase moves along. It exposes about 10 to 20 bases at a time, and it pairs new nucleotides to the existing DNA. And then you have termination, but it's different in prokaryotes and eukaryotes. The details of the termination are not that important. Now, we'll take a break on this one.